All right, seems like we're being recorded. If at any point you have any questions, please feel free to ask. I'm not sure how well I'll be able to handle them depending on what it's about because I'm not a creatine researcher. So we're going to skip past some of these introductory things. Like this, you're not going to learn this. I mean, come on, let's be real. All right. Here we go. So this is all basically introductory stuff. We're going to talk about some energy systems and bioenergetics. I'm assuming that everybody's had Phys, at least as an undergrad. I'll say everybody. There's only 2 people here. Have you all already had Phys?
Yes, sir.
All right,
Yes,
so you're in the graduate program. Is this the first Phys you take in your graduate program, or is there one before this?
For me personally, this is going to be the
Okay.
first.
So I'm actually an adjunct teacher. I'm in Texas at another university, and so different programs will call things differently. Like at LSU, there was the graduate Phys, and then there was the advanced graduate Phys, different courses. So that's where I got my doctorate, is LSU. Okay. So bioenergetics. I actually like this topic. It's one of my favorite ones to teach in the undergrad class. 3 main systems: your immediate, or the creatine phosphate, your glycolysis, which you could consider like an intermediate system. It's still fairly short, fairly fast. You get a decent amount of ATP, not as fast as creatine phosphate. Can't keep it up forever, which is why you have a long-term aerobic system. We're going to get into details of all of these. I'm kind of afraid to even expand the screen because it might disappear on me. So I'm going to leave it like this. Okay. You look at glycogen. You don't actually burn glycogen. And you don't actually burn glucose. So when you undergo glycolysis, you actually burn G6P. So whether you have glucose in the blood or glycogen stored in the muscle, it has to be converted into glucose 6-phosphate, which is right here, in order to enter glycolysis at all. So the glycogen, you're in a series of glucose molecules connected, stored in either muscle or liver, and of course glucose is a single 6 carbon molecule in the blood. Whenever it's moved into a cell, it has to be converted into G6P by hexokinase. Similarly, glycogen breakdown, you have to convert from glycogen to G1P to G6P. So these things take a little bit of time, and if you need energy right away, it's faster just to immediately take the ATP. So ATP is pretty big, so it's stored in fairly limited quantities in the body, in the muscle specifically. So we're talking about exercise, so we're going to talk about muscle. And you never actually deplete ATP, no matter how hard you're working. Oh man, I'm hearing people in the hall. Okay, so any reason, any explanation that you can give as to why you don't deplete ATP, no matter how hard you're working? Anybody? Of the 2 people? It's more fun if it's interactive. Can you think of any reason why you wouldn't fully deplete the ATP in your muscle when you're exercising?
205. Once they're ready,
All right, well, I'll cut to the
last
chase.
minute, then you're going to tell Russell.
You can tell I'm not alone.
They're going to empty it out, and then they're going to take
Hang
it
on.
to the fridge. And that. Okay, that goes too. Are
Okay.
you taking this after all?
It's a UTB thing, so I can't fit it, but Yulun said he would take it in his lab.
Who did?
Yulun, Todd.
So Ty wants it in his lab?
Yeah.
You heard.
All right, folks.
Okay. No.
All right, McNeese, are you still with me?
Got it?
Yes, sir.
Okay, good.
Yes, sir.
So anyway, you never really deplete the ATP in your muscles. You can't. You would be dead, really. And you always have to replace it using one of these bioenergetic systems. So the fastest one to replace the ATP is the creatine phosphate system. So it's literally creatine and phosphate bound together. So you got creatine phosphate right here. And you can actually break it apart to form ATP. You can also take 2-ADP and form an ATP and an AMP. When I say it's the myokinase reaction, the name for it. And AMP on its own will actually stimulate more glycolysis. So the more you break down ATP to ADP to AMP, the more stimulation you have to increase ATP being made. So you don't really run out of ATP. You're replacing it, and you'll actually fatigue before you can ever run out of ATP completely. You might go down to like 50% stores in your muscle. But it's big. Like I said, creatine phosphate is substantially smaller. So you can fit a lot of creatine phosphate in the muscle and use that to replace ATP pretty quickly. Okay. Usually when I do these lectures, I have a slightly different format, but that's okay. So you look at exercise intensity. This is kind of an overview of bioenergetics pathways, how they work. Exercise intensity and then substrate oxidation. You've got carbohydrates in red, lipid or fat in green, and then here's your RER, or your relative exchange ratio. It's a ratio of the oxygen consumed to the CO2 expired, and we can use an indirect calorimetry or metabolic cart to measure RER to then calculate carbohydrate and lipid oxidation. So with higher intensity exercise, you're relying more and more on carbohydrates and substantially less on lipid. Does anybody recall from their Phys classes why very high intensity exercise is going to pretty much inhibit lipid oxidation? This is why in-person classes are better. I can actually make eye contact and make someone feel uncomfortable enough to answer. No? Okay. Well, we'll get into it more, but high intensity exercise, you're relying a lot on glycolysis. So when you're using a lot of glycolysis, what are the two products that you might make from glycolysis? Either Brandon or Sierra. Breakdown of G6P through glycolysis should result in formation of something major. You don't recall? Okay. Pyruvate would be ideal. But when you exercise at a high enough intensity to where you can't use the pyruvate fast enough, the NADH has to get rid of the hydrogen, so it dumps it on pyruvate. So pyruvate accepts 2 hydrogens and it becomes lactic acid. And an accumulation of lactic acid actually suppresses lipolysis and stimulates what we call reesterification, which is taking the free fatty acids and putting them back into the adipose tissue. So that's why at very high intensity you have a sharp drop in energy coming from fat. And of course, a big increase in energy coming from carbohydrates. Now you look at duration, it's a different story. RER should be relatively constant if you're going to have a 2 or 3 hour activity, like a marathon, for example. And at rest, you're going to be burning a lot of fat and relatively little amount of carbohydrates. But as soon as you start to exercise, your energy needs become immediate. So if you just start running at 7 miles an hour, it doesn't matter if you're not warm or not, you need a certain amount of ATP. So you're going to be up here. I'm assuming you can see my cursor. Can anybody see my cursor? Is anybody still awake?
I can't see it on my end.
That's too bad. All right. Yeah, I don't see it on my recorded screen either. So anyway, it'd be like 70%, I guess that is. What was I just saying before this? Okay, so if you jump on a treadmill, for example, at 7 miles an hour, your energy needs are whatever they are to run 7 miles an hour. It doesn't matter if you're warm or not. So you're getting that ATP from somewhere. It has to happen fast. So initially it's going to come from creatine phosphate and stored ATP, but just for a couple seconds. And then your glycolysis system is going to speed up and then provide the ATP that is needed while your oxidative system then gets moving. So The oxidative system is the biggest one, most moving parts, takes longer to get moving. Takes a lot of ATP, but at the start of exercise, it takes a while to get moving. Now this might be a little bit misleading, showing, all right, lipid is going to come up immediately and carbohydrate is going to go down. I mean, it's kind of true. What it'd be nice if they would show is aerobic versus anaerobic, or oxidative pathway versus anaerobic pathway. That's probably a better way to do it. So we can pretend like the carbohydrates are anaerobic pathway just for a second. It's going to increase immediately while your aerobic one, at least the percent contribution, decreases. You get your aerobic system kind of warmed up, and it's going to increase pretty quickly. That's how I would like to start this lecture. But we're talking about substrate oxidation. So the longer you're exercising, the more you're going to shift from burning carbs to burning fat. This shift from carbs to fat is going to be even more pronounced the more fit you are. So this person's example is obviously very fit, because if you're not fit, you're not going to exercise for 3 straight hours, basically. So you're better at oxidizing fat. You can start burning fat sooner, increase it the entire time. As time goes by, you're going to decrease the amount of carbohydrates you're oxidizing. And a big reason for this is because the brain is pretty greedy. The brain and the spinal cord rely almost exclusively on carbohydrates for their fuel. So when you're exercising, you're going to use carbohydrates stored in the muscle first. So exercise or bioenergetics is kind of like real estate. It's location, location, location. If you're running, you're using your legs. So ideally, you're going to use the fuel that's stored in your legs first. That includes carbohydrates, fat, anything. So as you use the carbohydrates that's stored in your muscle, you're going to have to replace them with the glucose that's in the blood. Okay? No problem, except when the blood glucose drops, the brain kind of says, look, I need to eat too. So can anybody tell me where the carbohydrates would come from in order to increase the blood glucose at this point during exercise? This is still kind of like a refresher from undergrad. Undergrad meets grad. So you're using the carbs in the muscle, they're replaced by the glucose inside the blood, and that gets to a certain level. You need to increase the blood glucose, and that comes from the liver, because the liver has an excellent reservoir for storing carbs as glycogen. It's very energy dense. So you're going to release the glycogen from the liver, put it into the blood so you can maintain constant blood glucose. But everything is finite, and if you're not constantly adding glucose back into your system, then you're going to run out of carbohydrates. So you're going to have to switch to burning fat. Now, if you're not adding carbs to your system because, let's say, you're on an Atkins diet, well, that introduces a whole lot of other kind of biochemical problems. You recall the phrase, fat burns in the flame of carbohydrates? Yes, sir. Okay, good. You can't burn fat without burning carbs. So when you break down pyruvate and convert pyruvate to acetyl-CoA, you're also going to make oxaloacetate, which is a required intermediate in the Krebs cycle. And the Krebs cycle is really the first major aerobic process that happens. So you literally have to burn carbs in order to burn fat. And if you don't have carbs, then you have to break down protein to get the amino acids. You can take those and you can either build carbohydrate to break it down, or you can build pyruvate, you can build acetyl-CoA, you can build the intermediates for the Krebs cycle. So there's a lot of things you can build. The point is you're losing valuable muscle tissue when you do it. So this shift we're talking about in B, figure B right here, it's a lot more pronounced if you're not loaded with carbs or if you don't carb up. You're running that marathon, your carbohydrates are going to drop pretty quickly if A, you're on a ketogenic diet, or B, you don't drink like Gatorade or do the goo packets or whatever. So it's going to drop down to near zero levels. Say near zero because, like I said, you'll break down the protein in order to continue with glycolysis. So you look at various types of exercise. 100-meter sprint, it's very anaerobic, and for most people it's very fast. I am not a fast person, so I look at the 100-meter thing and think, all right, there's like 17 seconds out of my life. So this ratio might be a bit different for people with predominantly slow twitch fibers like me. So if you're really good, 10 seconds, you're done. So you're going to rely a lot on ATP and creatine phosphate, and then also some on glycolysis. But then you get into something like rugby or football, lacrosse, soccer. That's a lot of stopping and starting. So you're still doing a lot of creatine phosphate and ATP, but it's more than just a few seconds at a time. So you have to rely a lot more on glycolysis. And since you're going for really a couple of hours, you're using ATP, so you replace it with creatine phosphate, but then you have to replace the creatine phosphate with something. How do you build creatine phosphate again? Well, you have to use ATP. But you just made the ATP with the creatine phosphate, so we have a dilemma here. You can't pass the phosphate back and forth between ATP and creatine phosphate. So that's where glycolysis steps in. Glycolysis can make ATP to replace the ATP, and also to make enough ATP to replace the creatine phosphate. So that's a very big role for glycolysis. All right, you get into marathon running. Okay, very little immediate system is required here. Some carbohydrates are used, but they're used predominantly aerobically. So if you look at carbohydrates and fats, the green should say aerobic. You can use carbohydrates, fat, and protein aerobically, but anaerobically, you're basically only using creatine phosphate or carbohydrates. So in your mind, you need to relabel this to say aerobic is green, anaerobic is gray. That would make more sense. Energy sources for muscle as a function of exercise duration. Okay, you've got oxidative, non-oxidative, and immediate. Well, obviously immediate, your creatine phosphate system happens immediately. So the immediate contribution right here drops off very quickly. Within a few seconds, you're not really contributing anymore. Non-oxidative, which is, I've never seen anybody write it as non-oxidative. Usually they just write anaerobic, and we're talking about glycolysis. Speeds up very quickly, and then it eventually comes down as you're picking up your oxidative system. Now you can, like I said, you can break down carbohydrates or fat oxidatively, and those are the preferred methods. You can also break down protein, but it's not preferred. So we're still doing kind of a basic overview of the three energy systems. What energy storage capacity? Your ATP, creatine phosphate, you can't store that much, but you can get energy from it very quickly. Rather than calling this lactic acid, you really need to call it glycolysis, because glycolysis makes lactic acid. And depending on your muscle size, you can store a fair amount of glycogen. So it's stored in the muscle as glycogen. Muscle and liver is glycogen, and then the blood is glucose. And it's fairly quick, not nearly as fast as having it ready in ATP's format. And then aerobic, there's really no limit to how much you can store for the aerobic pathways, since you can use carbohydrates, fat, and protein. You can run out of carbohydrates, but I don't think a person can really run out of fat and be alive. It doesn't start quickly. It takes a few minutes to really be in full effect, depending on your fitness levels. So you have to rely on the other two at the start of exercise, even if it's a low-intensity exercise. If you go from sitting to a light jog, you still have to rely on creatine phosphate and glycolysis initially until your aerobic system speeds up enough to provide all the ATP necessary. Now, when you're doing strictly aerobic stuff, it's not, they say it's difficult to really deplete. I'm going to say it's not possible to fully deplete because you're also using fat. Now, we have a few slides talking about different bouts of activity time. So 0 to 20 seconds. Now, to be clear, we're not talking about any intensity level you can think of, because I can walk for 20 seconds very slowly, and this illustration is not applicable. So what we're talking about here is the maximum amount of exercise you can do at a very short duration, up to 20 seconds. So you're starting off with creatine phosphate and ATP right away. It's providing most of the energy needed throughout at least half of this. Your anaerobic system, which is glycolysis, is going to start to kick in, but honestly, it doesn't have that much time to kick in, between 0 and 20 seconds. You're not even going to see your aerobic system even blink in this amount of time. 20 to 80 seconds, all right, you're going to actually see some realistic contributions from glycolysis, and you might even start making some lactic acid. Still have to rely on creatine phosphate, ATP initially, and I'm going to disagree with the purple line here. Even in 80 seconds, well, okay, if you're very fit, like a very well-trained marathon runner, you can see a significant increase of your aerobic contribution in this amount of time. If you're unfit, like your sedentary average Joe, it's going to take more like 4 minutes to get your aerobic system up and running to full extent. So there's a big variation there, and we really need to distinguish that in our minds. So the longer the duration, the more the aerobic system is going to separate itself. So if you're going 200 seconds, all right, big contribution from the aerobic system. You don't even really notice creatine phosphate contribution at that time. All right. ATP turnover rate. This is just another way to say how fast can you use these substrates to make ATP. Your phosphagen system is the same as saying creatine phosphate, and it's very fast. So you can make ATP extremely quickly. Glycolysis, it's also pretty fast, not in the same ballpark. Oxidatively making ATP from carbohydrates, right? And that's taking the product of glycolysis, which is pyruvate, converting it to acetyl-CoA, having it enter the Krebs cycle, making NADH, FADH, GTP, sending the electrons and hydrogens to the electron transport chain, so they can pass through the various pumps and generate ATP. I mean, that whole process, that's what we're talking about for mitochondrial carbohydrate oxidation. It includes glycolysis, which is anaerobic, and then everything that starts the Krebs cycle and beyond, which is aerobic. So that's next in line, because it's faster to break down carbs than fat. And of course, the slowest turnover rate would be fat. Fat's the biggest particle by far. The most common one is palmitate, and that's a 16 carbon molecule. So it's common in the body because it's common in the diet. And that's pretty much what every example is going to use when you talk about free fatty acids. 16 carbons, so you keep having to break off a 2 carbon chunk so you can convert it into acetyl-CoA. Then the acetyl-CoA goes through the Krebs cycle, electron transport chain, etc. So it's a bit of a process. So just to get ahead of myself a little bit, whenever you take a free fatty acid, you break down this free fatty acid, you break off a 2 carbon chunk and make acetyl-CoA. That's called beta oxidation. Some people mistake beta oxidation. They think, okay, breaking down fat to make ATP. That's beta oxidation. No, that's not. That's oxidative phosphorylation. Beta oxidation is very specific to breaking off a 2 carbon chunk from a free fatty acid and converting it into acetyl-CoA. I like figures as study guides because they can give you a lot of information. So we should use this as something to reference whenever we go through the testing material. By the way, whenever I'm going to give a test, it should be pretty obvious. It's an asynchronous online class, so it's not like you're going to show up and take a written test. So test or take home. I mean, to me that's obvious, but I figured I'd just say it. What I'm going to focus on really in the tests is to see your level of understanding and be able to put things together to have a coherent answer showing, mastery of material, rather than, give me the name of an enzyme or what metabolite becomes another one. So you guys are going to be able to look up all kinds of details, but the more important thing is to be able to put it in context. All right, that being said, at rest, you have a lot of ATP. When you're mildly fatigued, you still have a lot of ATP. Severely fatigued, your ATP went down quite a bit. And severely fatigued just means you did some hard work. prolonged fatigue, we're talking like marathon running here, you still have a little bit less ATP than at rest, but not much because you're making so much ATP through fat oxidation. All right, now ADP, that means you've already broken down ATP once. The triphosphate's now diphosphate. So at rest, you can see the concentrations are lower than during mild fatigue, which makes sense. You break down ATP to work, and work can cause fatigue. So that's why ADP concentrations go up as fatigue goes up. Now you notice the 0.13 for severe fatigue is substantially lower than prolonged fatigue. Well, that's because you are able to make a lot of ATP during a marathon, and one of the ways you can do that is to combine two ADPs to make an ATP and an AMP. So you can clear the ADP fairly quickly while you're making new ATP. So AMP, all right, at rest, it's negligible. Mild fatigue goes up a little, still negligible. Severe fatigue, you can finally see some concentrations. And again, prolonged fatigue, it goes way down again because you're clearing it. You're actually using it. There's no such thing, in my opinion, as a metabolic waste product. AMP is an example. Some people would call it a waste product, but it also stimulates glycolysis. So it's not really a waste. It's just used for a different thing. Now, I'm not going to go through all of these things, just a couple of the ones that I think are going to stand out, like creatine phosphate. At rest, it's high. Mild fatigue, fairly high. Severe fatigue, very low. That means you are doing a very hard workout. If it's very hard, it means it's very intense, so you have to rely on getting ATP very quickly, which is creatine phosphate. A marathon, we saw in the previous slides that for long duration exercise, you basically are not using creatine phosphate much at all. So lactate, low at rest, comes up with mild fatigue, goes up a lot with severe fatigue. So again, just to touch base, fatigue happens with exercise. If it's mild fatigue, you're exercising a bit. Severe fatigue, you're exercising very intensely. Prolonged fatigue, you're very aerobic, and so you're not making much lactate. Your pH should be fairly consistent, except, of course, when you're doing very high intensity work. That's going to change for a few reasons, which we'll get into in more detail later, but when you break it down ATP, you're going to release energy, high energy phosphate. It's going to be released. You're also going to release hydrogen, and the more hydrogen you add to the mixture, the lower the pH is going to be. All right, so glycogen. This one's actually a little bit different than the others because it goes down with every single category. Highest at rest, goes down with some exercise, goes down more with heavy exercise, goes down the most with very long exercise. There's an example of ATP. All right, I hear people coming back again, so I'm probably going to have to shut this lecture down soon. We'll find out. Give me just a second to close the door right next to me. Okay. I'm tucked away behind as many doors as I can find. All right, so ATP is very large, and you have two different places you can cleave ATP to ADP to AMP, so you can release phosphate and energy. So two sites where you can release energy. Adenosine triphosphate to diphosphate to monophosphate. And since it's so big, you have a very limited quantity that you can store in any particular gram of muscle, which is why we have to rely on a creatine phosphate. Okay. This looks like a really confusing slide because of all these strange terms, right? It's not that confusing, to be honest. All right, you make ATP from the mitochondria, or you make it, say, with glycolysis. What's the production rate? All of these different things are cells. They're just different kind of cells. Okay, so certain cells are better at making ATP aerobically. Some cells are better at making ATP through glycolysis. So that's really all I'm going to say about this. This just gives you an overview. You've got the muscle on a microscopic level at the bottom. So these are pictures of sarcomere. And you're using ATP for the power stroke. becomes ADP and has to be replaced so that your muscle can relax. So you're going to have to replace that ADP with new ATP. Well, where are you getting it? You can get it in the mitochondria right here from creatine phosphate. It can be made in the mitochondria. You have oxidative phosphorylation that happens in the mitochondria. You can make creatine phosphate to replace the ATP, and the ATP that replaces the ADP will cause relaxation of the myofibrils. Now, I mentioned that the previous lecturer for this focuses a lot on creatine. So we're going to cover it, but not in the same detail that he would cover it. And at the same time, when we get to other aspects of bioenergetics and also vascular, we're going to cover that in more detail than maybe the slides would show, because that's more my thing. All right, so you can convert glycine to orthine in the kidney. It's going to travel to the liver, where it'll then be converted into creatine. And the creatine is going to have to accept a phosphate from ATP. So ATP is going to donate a phosphate, add it to creatine, so it becomes creatine phosphate. And that's then going to be stored in the muscle until you have to use it to replace another ATP. So it'll donate its phosphate to ADP, then converting it to ATP. And then you're left with creatine again. So, but the creatine doesn't go to waste. I mean, eventually everything has to be replaced. But you can use the same creatine many, many times. So this is going to go right back in to this cycle. I keep forgetting you can't see my cursor. All right, the creatine on the left side is going to go right back to the same cycle where it's going to accept a phosphate from ATP and then keep being used. And the cycle just keeps continuing. So you can eat creatine. It's a supplement. You can make creatine, so it can be absorbed from your intestines into the body. If you consume it, you can then. Some people don't actually have any benefit from consuming creatine phosphate because they're already making as much as they can use, and that, of course, will happen on the right-hand side where you're talking about liver and kidneys. You can take different amino acids, like arginine, glycine, and methionine, and then just make all the creatine phosphate that your body needs. So those are the main ways you're going to get creatine phosphate in the body. Supplements of actual direct creatine or just making it. And how do you get rid of it? Well, kidneys. Creatine is kind of like glucose in that it attracts water. And on the flip side, if you want to get rid of it, you're going to use osmolality as a way to filter out some of the creatine. So it's going to lead through the kidneys. So you can also break it down in the liver. All right, we're back to ATP. Oh, sorry about this. Let me hit this button. Okay. So there's your ATP. You've got your high energy bonds. You can break them down with the enzyme ATPase. I personally think this enzyme is the best named one that I can think of. I mean, you know exactly what it is. It ends in ACE. It's an enzyme. Breaks down ATP. Sometimes when I ask my undergrads what's the enzyme that breaks down ATP, nobody can answer. All right, hold on a second.
Oh, the fridge is empty.
No, you need it now?
Got it.
You ready?
No. That's fine. No, it's just like about 30, 40 minutes.
Okay.
Yeah, like 3 minutes away.
Okay. Sorry, it's going to get louder in here for a second, so I'll probably have to speed things along and then just cancel the rest of the lecture. All right. ATP phosphocreatine system. I kind of feel like we've talked about this a lot. It's really not that complicated if you break it down. You use ATP, you replace the phosphate with the creatine system. Okay? And this slide pretty much summarizes every molecular aspect of that conversion process. Here's the nutshell right there. Okay? You want ATP? Okay, you have phosphocreatine. It donates a phosphate to ADP to make ATP. Then you're left with free-floating creatine. All right, this is a big change of direction. We're talking about basically muscle contraction velocity here. You have fast twitch fibers, slow twitch, intermediate, and it's based on biochemical properties, and one of the most robust is what kind of isoform of myosin ATPase you have. Type 2X has a fastest myosin ATPase isoform. So what it does, it breaks down ATP from the myosin. When that happens, you have the power stroke. And the faster you break it down, the faster that twitch or the power stroke can happen. The slowest one, type 1, has the slowest myosin ATPase. I usually tell people you want to learn the biochemical properties of the fast twitch and the slow twitch, and then just know that type 2A is intermediate, because it's kind of in between those two. So the fastest twitch fibers have the fastest isoform of myosin ATPase, which makes the contraction velocity literally faster than the other ones. So when they say a fast twitch, that is a literal statement. A muscle twitch happens substantially faster if it has fast myosin ATPase. Okay. Now, like everything else that happens with bioenergetics, you have to transport creatine phosphate as well as carbohydrates and fat. And so you talk about the shuttle inside the mitochondria and the membrane. Again, like I said, I'm not going to go into much detail about this right now. One reason is because it's very loud in here and I'm having a hard time concentrating. My apologies. All right, supply and demand. So bioenergetics is a lot like a business course. So ATP consumption is demand, ATP supply is the supply part. So which side is winning, supply or demand? Depends on the exercise intensity that you're doing and how efficient your bioenergetic systems are at replacing ATP. So we covered which kind of systems are fastest to replace ATP. So creatine phosphate is by far the fastest way to do it. If your exercise duration is very short, then your supply and demand is perfectly met. You're talking like 3 or 5 seconds. You won't even see any real change in ATP because you're going to replace it immediately with creatine phosphate. The longer the duration of the exercise, the more the mismatch between supply and demand. Just got to say, I don't know how much you can hear where you are, but having people using power tools to take apart stuff in the lab and in the next to the lab where I'm sitting right now is really distracting. Can you guys hear all that noise?
I actually can't hear it.
You're lucky. It's really loud and annoying.
I hear them coming in every now and then. But
Yeah, I keep closing the doors. I mean, there's a series of like interconnected rooms, and I strategically placed myself in the most interior one so I can have more closed doors, but they're working their way in. What can I say? This was all supposed to have been done a few weeks ago. So again, my apologies. Okay, where do we leave off? All right, ATP-PC system. It's just strange for me to have this much time dedicated to the creatine phosphate ATP system. It's the simplest one in the body by far. Now, hopefully you can remember some of this from your undergrad days. All right, so what enzyme is responsible? Well, creatine kinase. If you recall from your undergrad days, I mean, let's be honest, you probably don't, but different kinds of enzymes are named in different ways. A kinase is a class of enzyme which moves a phosphate. So creatine kinase will literally move a phosphate on or off creatine. So it's going to either build creatine phosphate by taking a phosphate from ATP, or replace ATP by taking the phosphate from your creatine phosphate. Okay. There's what I just said. Creatine phosphate plus ADP Using creatine kinase makes creatine. You have a couple of ADPs, that's the adenylate kinase, or you can use AMP and hydrogen with AMP deaminase, I swear I can say that, and you're making ammonium, not ammonia. See, ammonia versus ammonium. Why is it important? Low-dose ammonium preconditioning enhances endurance and submaximal physical exercises. So whenever you are making more ammonium, you can increase endurance. So now we're getting into a lot of like the nitty-gritty that your previous employee focused on. So his research focus had a lot to do with creatine. So, I mean, I'm going to skip past some of this stuff. How's it work? I mean, we've said it about 45 times so far. Okay. Now there are some, this is where you can really tell he's really interested in creatine. So this is a really good lecture for anyone interested in creatine. The effects of creatine supplementation on thermoregulation and cognitive performance. So it is potentially therapeutic. Creatine supplementation is potentially therapeutic for several things, including right here, cognitive function, as well as quality of life for dialysis patients, reduces cardiomyocellular injury in people with heart disease. And this one we're talking about neuronal cell death, how it relates to both exercise performance with motor neurons and also dementia. Cell death and also cell dysfunction in the central nervous system is largely responsible for dementia. And some of these processes that create like these amylase coatings on your neurons can be inhibited with sufficient creatine phosphate, or at least in theory. None of it's really proven yet, but it's interesting theory that creatine phosphate can slow the progression of neuronal death and impairment. Still the same topic right here. Alzheimer's disease, amyloid bodies are coating some of the spots in the neurons in the brain, causing a much slower firing rate. And the theory is that creatine supplementation can alter like the leakiness of certain parts of the neuron, which can slow down the buildup of these amyloids and possibly slow down the progression of Alzheimer's disease. Same with Parkinson's disease. And this is original work, not by me, but by the previous lecturer, and it shows that there's a possibility that creatine supplementation can reduce headaches and dizziness. Fatigue, to me that's the most obvious. So we're not talking regular headaches here, we're talking like migraines, because migraines are going to happen with the rapid contraction and relaxation of the precapillary arterioles inside the brain, causing an imbalance of pressure and then severe headaches. And you can potentially reduce this effect with creatine supplementation. But this is not my research, so I'm really not going to go into a lot of detail about that. All right, really any detail about that. All right, basic glycolysis. I think that was more than enough attention spent to the creatine phosphate system. I mean, that's the shortest, fastest, most simple of the three bioenergetic systems. Okay, basic glycolysis, obviously start with glucose. You don't break down glucose in glycolysis. Does anybody remember what I said in the start of this lecture? What do you use instead of glucose to enter glycolysis? So you're both at a disadvantage because it's not like there's 30 people in this class. There's only 2 of you right now.
Did you mention the carbohydrate usage for this process, or is this?
Yeah, I'm talking about the carbs, but it's not it's not glucose that you're actually breaking down in glycolysis. It has to be converted to something else first. You recall what that is?
Pyruvate?
No, pyruvate's the end result. So you're just a little bit ahead of where I am right now.
A little rusty.
Yeah, it's fine. I mean, I teach this kind of stuff every semester, and every time the semester starts, I'm rusty. You know, give me a couple of weeks, then I get in my groove. All right, so you break down glucose or glycogen to form glucose 6-phosphate. So G6P. That's actually what enters glycolysis. And from there, that's where you got a little bit ahead. You make pyruvate. If you can't really keep up with the pyruvate production oxidatively, so the pyruvate is supposed to be used oxidatively in the Krebs cycle, also known as the tricarboxylic acid cycle or the citric acid cycle. It's all the exact same thing. If you can't use the pyruvate as fast as you're making it, it accepts a couple of hydrogens and is converted into lactate. So the harder you're exercising, the more hydrogen you produce, and the more NAD and FAD has to shuttle those hydrogens around. And if you make it so fast, the hydrogen so fast, they don't have time to travel all the way to the electron transport chain. They're going to dump it off right there on pyruvate, making lactate. Which is why when you exercise harder, you're making lactate faster. So you literally have to free up the NAD and FAD just to grab more hydrogen so glycolysis can continue. If you don't have NAD and FAD, glycolysis just stops. So lactate, in a way, kind of helps keep you exercising longer. It also, believe it or not, helps buffer pH. So just now I said that pyruvate accepts 2 hydrogens to become let's say pyruvic acid accepts 2 hydrogens to become lactic acid. Well, lactic acid donates one of those hydrogens back into the milieu to become lactate, and that's why it's considered an acid, because it donates a hydrogen. So you might think, all right, it's an acid, it makes things more acidic. And if you just look at that one snapshot where lactic acid donates a hydrogen to become lactate, then you'd be right. But back up a step, it took 2 hydrogens on and then only gave 1 back. So really, producing lactate in the long run, in the big picture, actually helps buffer your pH, because you're minus 1 hydrogen in the grand scheme of things. You took 2 on to convert pyruvic acid to lactic acid and only gave 1 back to make lactate. So lactic acid buildup doesn't actually happen. It's lactate buildup, and it's there to help buffer your pH among like dozens of other things. Lactate's a really interesting molecule. All right. The biochemical reactions involved with carbohydrate metabolism, some of these are really not going to be applicable to exercise, to be honest. So glucose, number 2, glucose makes 2-pyruvate, as well as 2 NADH, 2 ATP, and 2 waters. Well, that's just glycolysis, so it's very applicable. And then you go to step 3, you take pyruvate, can make acetaldehyde, or CO2. You look at number 4, pyruvate can make ethanol. All right. Well, these are not traditionally what you think of as aerobic. You go to number 5, pyruvate plus CO2 with carboxylates makes oxaloacetate, which is required for the Krebs cycle. So you have the Krebs cycle, which is the first part of aerobic metabolism. You can also have gluconeogenesis to make the pyruvate back into glucose. So there's a lot of steps. I'm not going to go through all of these because each one is its own complex reaction. So just looking at a reaction, well, let's be honest, looking at 12 reactions on one slide is not really going to give you an in-depth idea. So if you didn't know, C6H12O6 is glucose. NADH, it's the reduced form of NAD. There you go. TPP, thiamine, prophosphate. GSH is the reduced glutathione. GSSG is the oxidized glutathione. G6P, which we talked about, glucose 6-phosphate. F6B, fructose 6-phosphate. 6-phosphate. I swear I can speak. I'm teaching another class at the university where I'm full-time, and it's going to be an interesting challenge. It's all about muscle biochemistry for exercise, and I have taught it before, but it's been like 6 years. And just seeing fructose 6-phosphate is reminding me that I'm about to teach that. All right, comparison of glycolysis. This is such a misnomer. All right, aerobic glycolysis doesn't exist. It's a unicorn. All right, there's no such thing as aerobic glycolysis. Anytime you use the word glycolysis, it is always anaerobic, period. That doesn't mean it happens in the absence of oxygen. There's only one thing that happens in the absence of oxygen, and that's death. It doesn't require oxygen. That's the thing. What they're trying to say is aerobic glycolysis yields pyruvic acid. No. It's oversimplified. Glycolysis is anaerobic. It makes pyruvic acid, period. That's what happens. What else is going on inside the cell is going to affect what happens to the pyruvic acid. If you're making pyruvic acid so fast that your NAD and FADH can't keep shuttling all of these hydrogens and electrons to the electron transport chain, they're going to convert pyruvic acid into lactic acid. So the glycolysis is always anaerobic. It's the exercise you're doing that could make you more or less anaerobic. You slow down, you're not producing hydrogen as fast, so your NADH and FADH has a chance to shuttle the hydrogen to the electron transport chain so that you can actually use your pyruvate to convert to acetyl-CoA and go into the Krebs cycle. You're going very intensely, you don't have time for the NADH and FADH to make it all the way to the electron transport chain. They dump it off on pyruvate before it can even be converted into acetyl-CoA. You make lactic acid. Therefore, you're in an anaerobic state. All right, feeder pathways for glycolysis. I think this is a really chaotic looking slide. Honestly, the slide in the undergrad Scott Powers book, I think is much better than this one. But basically, you're going to get glycolysis from carbohydrates, hopefully mainly carbohydrates, and general metabolism. Sorry. Yeah, right here we have feeder pathways of glycolysis, and then you go into full metabolism. Okay. All right, feeder pathways of glycolysis. You can have fructose that can be converted in the liver to a usable function of glucose. Really, that's about it. Glycolysis is just glucose. So you can use glucose, you can use fructose, you can use amino acids that are then built into glucose, but you can't use fat. You can use any sugar. All right, metabolic summary here. When you exercise, you're going to use a lot of carbohydrates, no matter what intensity you're doing, because it's just fairly easy, and you can do it for a while. You're also going to end up, hopefully, using fat and lipid if it's more than just a couple minutes of exercise. So the fat and lipid is always going to be aerobic, no matter what. Carbohydrate metabolism can be aerobic or anaerobic, but it always starts with anaerobic, glycolysis. That's every time, that's how it's going to start. But if it continues to be aerobic, then you're going to produce pyruvic acid, acetyl-CoA, enter the Krebs cycle, or here it's labeled as the citric acid cycle, make a lot of NADH and FADH, which goes to the mitochondrial electron transport chain and generates a bunch of ATP. The fat and lipid is always aerobic, period. You have lipolysis, which is the breakdown of lipid into free fatty acid and a glycerol. 3 free fatty acids and 1 glycerol. Circulates, gets transported by CRAT 1 and 2 into the muscle and the mitochondria. You have beta oxidation, which is breaking off the 2-carbon chunk of fat to form acetyl-CoA. which then enters the Krebs cycle, and then from that point it's exactly the same as what we talked about for carbohydrates. All right, proteins, hopefully you're not using protein to a large extent for exercise. Because if you are, it means something is definitely going wrong in the body. Still, you can break down protein. It'll form a nitrogen pool, which is unpleasant. It makes breath bad, cognitive function decline, and urine stink even more. But you break down the protein to amino acids, and you can build whatever it is you need to keep going. Carbohydrates, you can build glucose. You can build pyruvic acid. You can build acetyl-CoA. So whatever you're missing, you can make with the amino acids. But if you think like you're a general contractor and you're ready to build something, and then someone says, okay, here's the plans, build it now. All right, let's look at the plans. What is it you need? All right, let's adapt. All right, if you have a lot of different things that you have to do, it might take longer. There's a lot of steps. So for this, you're talking about tearing down a building. Granted, this is not a perfect analogy, but it's pretty good. Tearing down a building in order to get the bricks and materials to then build a completely different building. It's a lot slower than just having what you need to build it in the first place. So it's not desirable because A, it's very slow, and it produces things like ammonia, which cause fatigue and can impair cognitive function. But here's a quiz for you. Where do you think the protein's coming from? What is it you're breaking down during exercise to get the amino acids? Come on, folks. Where do you have protein in your... you have protein everywhere, I admit. But let's say you're running a marathon and you run out of carbohydrates. Take a guess at where you think the protein's probably going to come from. No, that was the incorrect answer. Probably coming from skeletal muscle. So the thing that you're actually using is being destroyed. This is a pretty bad drawing, I have to say. This is a pretty bad cartoon of skeletal muscle structure and function. This one's a little bit better, but also pretty complicated. Skip. Here we go. This is where we were whenever we first started this lecture. Now you're talking about not really metabolism, but what's going on to excite the muscle. So you've got a presynaptic neuron, and you have these little vesicles which contain acetyl-CoA. No, not acetyl-CoA. Wow. I have a total brain fart right now. What is acetylcholine? There you go. Acetylcholine is the neurotransmitter that goes across the synaptic cleft in order to cause depolarization, opening up ion channels, etc. So you open the ion channels, it causes depolarization, which goes down the T-tubules, opens the calcium channels, calcium floods out. Does any of this sound familiar? So calcium floods out, binds to troponin, makes tropomyosin move out of the way, so you can have the myosin and actin interacting, and then a contraction happens. I'm not really sure why they're putting this particular slide here, because it's all about muscle excitation, contraction, relaxation. Okay. All right, this is really simple. Glycogen location. Where is it? In a nutshell, it's in the muscle, it's in the liver. That's where it's located. Okay. Sorry, it's getting pretty loud over here. They're like literally yelling.